A very good morning to all of you. I welcome all my pharma fraternity members to this platform. The platform is dedicated to make awareness of clinical pharmacy and cover the courses of the syllabus as prescribed by PCI for the clinical pharmacy and the pharma practice and patient oriented pharmacy related courses. In this slide, we will overview the therapeutic drug monitoring, and with this we have completed almost the syllabus topic of M.Pharm clinical pharmacy. And if someone wants to get a overview of all the topics right from the introduction unit 1 to unit 6 of PCI, he could go through the first slide till the 10th slide of this whole effort, and it would help you to frame good notes by blending this information from the books which you routinely refer. So मेरा ये प्रयास है कि इससे हम क्लिनिकल फार्मेसी के प्रति एक अवेयरनेस ड्राइव को इनिशिएट कर पाएँ, जिससे क्लिनिकल फार्मेसी की अवेयरनेस को स्ट्रेंथन मिले और आपको एक विज़न मिले कि हमने अपने नोट्स किस तरह बनाने हैं, कौन कौन से टॉपिक्स हमने किस पैटर्न में तैयार करने हैं, ताकि आप अच्छे मार्क्स अपने सेशनल्स और मेन एग्जाम्स में सिक्योर कर पाएँ. विद दिस आई वेलकम ऑल अगेन फॉर द ओवरव्यू ऑफ TDM मॉनिटरिंग. थैंक यू. सो दिस इज़ द फर्स्ट स्लाइड. इसमें हम इस टॉपिक को बैकग्राउंड और इसकी इम्पोर्टेंस और इंट्रोडक्शन, एपिडेमोलॉजिकल एस्पेक्ट, क्या इसका ओरिजिन एस्पेक्ट रहा, क्या इसका ग्रोथ एस्पेक्ट रहा, इस पे बात करेंगे. सो कमिंग टू द बैकग्राउंड, therapeutic drug monitoring, also referred as TDM, is a service. It is utilized for making the clinical decision to assist the dose adjustment on the basis of clinical and laboratory management. Usually, the blood concentrations are used to consider the dose adjustments and framing the individualization of drug dosing pattern of an individual patient. So, the basic importance is to frame a safe, effective dosage regime for a patient. It is a branch of clinical pharmacology. So in this we have defined it as epidemiology, rather than it would be a journey of this concept. So TDM was introduced initially in the 1960s, and on its first initiation step it was utilized for the epileptic patients. In the late 1970s, it was again utilized for monitoring and managing of ADRs and drug-related toxicity aspects. The main goal of this TDM approach is to do the dose optimization. The impact leads to the safe drug therapy through maintaining the blood drug concentration within the therapeutic range or therapeutic window. It is probably used for all drugs, but it is more concentrated for drugs which have a narrow therapeutic index, which means that there are fluctuations of their concentration beyond the range would manifest as negative out. So importance in other areas which is linked with therapeutic drug monitoring, or we are using this therapeutic drug monitoring approach in different areas like patient compliance, drug efficacy, drug interactions, drug food interactions, and individual response variations, or the toxic effects being manifested by the drug therapy. Overall, it assists in the therapy decisions either in initiation or in cessation. So overall, it helps to optimize the drug and making the drug therapy more safe and effective as per the clinical needs of the patient. Now in this slide we will see the process of TDM. इस स्लाइड में हम TDM का प्रोसेस देखेंगे कि क्या क्या प्रोसेस है जिससे हम TDM अप्रोच को इनिशिएट कर सकते हैं. तो बेसिकली देयर आर 5 स्टेप्स in which the TDM process is completed. The first is blood sampling, second is measurement techniques, third is communication of the results, fourth is interpretation of the results, and the fifth is the use of clinical decision making support systems or through the clinical judgment. So first we come for the blood sampling. Usually the TDM in the traditional approach uses the plasma blood drug concentration, in which the blood is the basic sample as the drug flows within the body in the form of blood dissolution or blood absorption process. So the drug is absorbed, excreted via blood. It is a carrier of drug and other excretras and other elements. Whole process of TDM and take it to the rational way, which includes the timing of blood sampling, the type of blood sampling, and the storage aspects, so that the consistency of the blood sample is ensured and the safe results are reported after the assay test or the lab test procedure. So next step comes after taking the blood sampling is the measurement techniques, which are different techniques used to assess the drug concentration in the blood sample taken. There are many techniques, usually chromatography, mass spectroscopy, ELISA, radioimmunoassays are used, and enzyme included gene therapy is also used. So there are many techniques which are used. The next comes the communication of the results to the team. So the basic is how we communicate the result to the team so that it could be interpreted easily and the data could be compared with other studies or other related information to make relevant interpretation. So after communication, the next step is to interpret the results. For the interpretation, there are many details which are required, like the dose regime, dose route, sampling details, assay characteristics. A blend of all these informations helps to interpret the result to the rightest of the accuracy to design a best suitable dosage regimen for the individual patient. Now, last step after interpretation comes the support taken for making a rational and the most accurate clinical decision. Level of database which they contain, and on the base of the increased data information, the more accurate and precise decision making support is given to design the required dosage regime for the patient. Now in this slide we will discuss the application of TDM. So continuing from the previous slide where we discussed the process of TDM, there are some key points which are required to be adhered to initiate or to complete a successful TDM process. So first is the correct dispensing should be ensured as per the prescribed information. Second, to make interpretation most accurate, the details of administration timing, the right sampling time, the details of assay procedure in the right way, the patient clinical details, comorbid conditions, concurrent details, and the information about the indications should be available to make a valid decision through TDM for designing the drug regimen for a patient. Now on the right hand side there are some areas where the TDM has shown its relevance. So the first important area is the serious toxicity that is manifested with some drug classes like anticonvulsants and antibiotics. So the TDM could help to decrease such serious toxicity events by monitoring of the plasma concentrations of the drug through comparing it with the standard required concentration which are required to give a suitable or rational therapeutic range, and it helps to prevent the concentration to reach the toxic level. So indirect way the serious toxicity outcome is inhibited or monitored. Now second comes some drug shows very steep response, different responses like theophylline. So to monitor those steep response dose curve, application of TDM is there. Previously it has been shared that for low therapeutic index drug in which a marginal change in concentration shows drastic effect in adverse effects or therapeutic failure. So the drug comes digoxin. Pharmacokinetic variability, because it is utilized for designing a dosage form for individual patients, so it covers the variability, the differences in responses either in pharmacokinetic or pharmacodynamic variability. It helps to alter the pharmacokinetic as well as the pharmacodynamic variability. It helps to improve the non-linear pharmacokinetics with drugs having marginal toxicity events like phenytoin, phenobarbital, lithium aminoglycoside. It helps to monitor drug interactions, drug food interactions. It is very useful to monitor drug therapy when it is used for a chronic use like long-term drug therapies for anticonvulsants in conditions or in ailments of epilepsy or diseases like sepsis. It helps to monitor overdose, and the example is acetamin toxicity which causes a severe event of liver failure. It also helps to predict the patient behavior and patient adherence pattern. Patient behavior includes the events of drinking and smoking which interferes with the drug doses of some classes of drugs. So overall, the application of TDM is to improve the drug therapy of the individual patient and design a more individualized drug therapy for a particular patient so that the rational use could be ensured and maximum safety could be conferred to the patient. Finally, we come to the conclusion slide. So in conclusion, the TDM is an important service. It helps to improve the rationality, it improves the treatment outcome, and the impact of the services could be improved by the use of software which could help to assist the practical decision making to make the drug designing or drug mixing it with the data of the book which you refer. You could make good notes and secure good marks in your sessionals or main papers. I thank my pharma fraternity for supporting me, and with this we have almost completed the clinical pharmacy course topics as prescribed by PCI. And if someone wants to overview all that, he could go right from the first slide introduction to the different aspects as described in the syllabus. तो इस स्लाइड से हमने क्लिनिकल फार्मेसी के जितने भी हमारे टॉपिक्स हैं पीसीआई के ऑलमोस्ट वो ओवरव्यू कर लिए हैं, और ये स्लाइड्स आपको हेल्प आउट करेंगी अपने कोर्स में क्या क्या चीज़ों को ओवरव्यू करना है, कैसे हमने सम अप करनी है चीज़ें, और कैसे हम नोट्स बना सकते हैं इनको मर्ज करके। ये आपको हेल्प आउट करेंगी। आप अपनी रूटीन बुक्स के साथ इन डेटास को मर्ज करके अच्छे नोट्स अपने लिए बनाइए और अपने सेशनल्स में, मेन पेपर्स में, एक्सटर्नल्स में अच्छे मार्क्स सिक्योर कीजिए और क्लिनिकल फार्मेसी को स्ट्रेंथन करने में अपना योगदान दें, so that we could make awareness and strengthen the sector of clinical pharmacy. थैंक यू, सभी को धन्यवाद, नमस्कार, प्रणाम।